Thank you, Rina, for that long introduction. I wish I would have kept my CV much shorter. So thank you also for the invitation to come on and speak on this topic, which is close to my heart and the subject, as Rina said, of my research. And I picked the title because I'm really not going to do a complete summary, but giving you including some very personal perspectives on the topic. So I always like to start with these slides because the fact that we discuss this topic at all, I think it's always important to stress that it's the result of our success with developing ART and providing increasing access to ART across the world. And that means that if you and that's really the result of access having preceded in the Northern Hemisphere over the Southern Hemisphere, the proportion of people with HIV living over 50 are increasing across the globe, and currently the proportion is highest in the Northern Hemisphere. But, of course, many more people with HIV live in the Southern Hemisphere, and therefore the absolute numbers of people living with HIV who are over 50 is much greater, including in your area of the world. So that's something to keep in mind. So we've learned, and this is just a summary slide, we've learned from the study that we've been conducting over 10 years now in Amsterdam, where we compare a group of people living with HIV, and these are almost 100% people who have been on long-term ART and well suppressed, to a group of people without HIV, and we selected those people from the STI clinic in Amsterdam in order to make them as comparable as possible to the people with HIV which come from our Amsterdam UMC HIV clinic. And we found that, generally speaking, and you see the various comorbidities listed on the slide, that comorbidities occur more frequently in people living with HIV. But, and that's an important comment to make, is that the differences that we found were smaller than what people were generally talking or speculating about. And all of that is really due to depending on which controls without HIV you choose. Because if your controls are very different, or if you don't know all sorts of risk factors for these comorbidities in your controls, but only in your people with HIV, you cannot appropriately adjust your analysis, and you may draw conclusions that the differences are much greater, and the wrong conclusion that this is all due to HIV, which is not the case. But even with our ability to control very, very carefully, the differences are there, and they are larger in people with HIV. We also found that the risk is very clearly multifactorial, and importantly that you cannot generalize for each and every comorbidity, and that the risk factors may be very different for different comorbidities. So, of course, lifestyle-related factors like for instance, smoking or substance use play a role. For cardiovascular risk, the known cardiovascular risk factors play a role also in the context of HIV. Body composition change is an interesting one because, like in the general population, for instance, with respect to frailty, there are relationships between changes in body composition and frailty. Co-infections, like for instance hepatitis C, play a role, and then something like systemic immune activation also plays a role. I think, and again, we can't generalize, but you can almost generalize. Certainly, having lived with untreated HIV and severe immune deficiency for a prolonged period of time really is a risk factor that one way or the other does tend to come out with each of these comorbidities. And then, of course, mainly older but certain ARVs, antiretroviral agents, may have particular toxicities, like renal toxicity, for instance, or bone toxicity, and that in itself may contribute to bone or renal disease. So one of the questions that was asked is the fact that they occur, that many of these comorbidities, which we know are related to normal aging, unfortunately that's the case, could it mean that the actual aging process is affected by HIV and maybe accelerated or accentuated biological aging? And I think from at least my take on data that have been fairly recently published and that I was partly involved with, The answer to that question, accelerated or accentuated biological aging, really depends on whether you look before or during ART. And I'll try to illustrate this. So this is just a schematic which I drew up together with Caroline Sabin for a review paper that we wrote on this topic a couple of years ago. So if you look at the top left, if HIV would have no, so here the y-axis is a marker of biological age, and calendar age is on the x-axis. So if HIV would have no impact on biological markers of aging, then the curves for those with and without HIV would overlap. If in panel B on the top right there would be accelerated aging, then the dotted line represents the acceleration in the people living with HIV, whereas the straight line is the people without HIV. So you would have acceleration over time. The curve would increasingly go up. Panel C on the bottom left is accentuated aging. So you would have one hit and then the curves would run parallel. So you would have a difference, but the difference over time stays the same, which means that there is, think of your basic physics, there's no acceleration. And D is if you would have a combination of the two. So why did I say that it depends on when you look? So this is a recent study from the Swiss HIV cohort study, which was very recently published in Lancet Healthy Longevity. And I think this was a group of 80 individuals that we studied, and I was involved actually with the study together with a person in my lab. If you look at the part of the panel which I encircled with the red, so if you concentrate on this part of the slide, you see that using epigenetic markers of aging, which at the moment in the aging field, I would say, if you think of in terms of biological markers, epigenetic signatures are the hallmark, and they're so-called a variety of epigenetic clocks, which we all studied and analyzed in this particular study, but I'm just showing you one of the clocks, which is the pheno-age clock. And they more or less, all of them showed the same pattern, that before people start ART, so to the left of the 0 on the x-axis, you see that the blue line goes up. So there seems to be an acceleration of epigenetic aging before antiretroviral therapy is started, and this is over a prolonged period of observation. So that study I just showed you is all people with HIV, so there are no controls without HIV in that study. This study is a study we did in Amsterdam, where we did have, like I just explained, these very comparable controls without HIV, but they're very comparable in all sorts of aspects, but they don't have HIV. This is a cross-sectional study that I'm showing you. So these are people with HIV who've been on ART for a prolonged period of time. They've been suppressed for a prolonged period of time. They have reasonable CD4 recovery, and we compare them cross-sectionally at one point in time when they entered the cohort to people without HIV. And we used a marker of biological age, which was developed by a European consortium outside of HIV, using a combination of various markers, 10 different assessments, including some, importantly, I think, the epigenetic markers that I mentioned previously for the other study. So here you see in red the people with HIV, in blue the people without HIV are controls, and you see a difference of some years in terms of biological aging. So this is accentuated aging. Think of those earlier curves I showed you. But the difference is much smaller than was reported in previous studies. And again, that has to do that the controls in those previous studies were actually inadequate controls to draw this conclusion. So it's an example of how you can draw wrong conclusions. What we also did in this study, we took a group of people with blood donors, and that's in green, and did the same assessment on those blood donors, and we find actually that those blood donors are younger than you would have expected for their calendar age, and both our people with HIV and our people without HIV are different from the blood donors. So the intriguing thing here is that there's also maybe something going on in this particular population at risk without HIV, which I think needs to be studied further. So back to the curve. So I would say that in the absence of ART, top right, Swiss study, there is evidence of accelerated aging. Whereas once you are on suppressive ART, the situation stabilizes and probably remains flat. Although formally we don't know that latter statement because we have not so far been able to study our group over time with these biomarkers. So ultimately the end conclusion is it's a combination. So what about, that's all markers of biological age. What about risk of comorbidity? Well, at least to my knowledge, I don't think there has been a formal assessment of the association between biological markers of aging and comorbidity risk. What we do know, of course, from various large studies, this is the SMART study that I'm sure you're all well aware of from many years ago, which showed that during compared continuous viral suppression to intermittent ART, so the virus would rebound, CD4 counts would decline, and once they declined to a certain level, people were started again. But that population, that group, was exposed to viral replication repeatedly. And the surprise was, but now we know more, the surprise was that certain severe complications which have to do with organ systems, so diseases that are normally related to aging, were more frequent in those that were intermittently treated, which fits with the concept that we have now. The same goes for a couple of years later, the START study, which looked at immediate treatment versus deferred treatment. Again, in the deferred treatment arm, in blue, there is a increased risk of serious non-AIDS events. So at least it proves the point that having replicating virus is probably associated with comorbidity risk. But as I said, whether that links to markers of biological age, I don't think has been studied yet. These studies, by the way, which have an enormous repository of samples, would actually be able to answer that question. So in our study, what we have been able to look at is what about the long-term evolution of these comorbidities? So is there an accelerated development of comorbidity during suppressive ART? What we find, concentrate on the red and the blue lines, they run parallel. So in this population, there is no acceleration of comorbidity development. As I showed you earlier at the beginning, the people living with HIV in red have a higher comorbidity prevalence, and that prevalence remains higher over time. But the curves do not divert. So there's no acceleration. We did find that for every additional incident comorbidity, the risk of mortality was about threefold. So there is an impact. We also looked at disability-adjusted life years development over time, and there we did find that the curves divert. The left-hand part of the slide, the HIV positives in blue, you see that the curves divert, but when we accounted for excluded years of life lost, so we took account of mortality, the curves no longer diverged, and that likely was due, not sure, it comes later, I'll go back to that in a second. Or I may have actually skipped this slide. No, I'll get back to that in a minute. So coming back to the Swiss study and this pattern, here if you now concentrate on the right-hand part of the slide, you see that once ART is instituted, epigenetic aging decelerates and then remains more or less stable over time. Again, there are no controls, and whether that pattern over time is still more biological aging than in people with HIV on suppressive therapy, will need longitudinal studies to tease that out. What I found interesting, and one of the reviewers of this paper actually picked that up, is that if you look at this spaghetti plot and you look at some of the individual curves, you see that there are individual differences, right? So the blue line shows that on average there's deceleration of biological aging when you institute ART. But if you look at the ones that I pointed out with the three red arrows, there are some individuals who have continued increased biological aging even after ART. And I think this is an area, so this is one of my perspectives that I mentioned in the title. I think this is an area that needs more work. What are the individual differences? Because what I have always found striking clinically in people who are on suppressive ART and do well in terms of viral suppression and do well in terms of CD4 count, clinically there are those people that deteriorate and become frail, etc., and there are other people that are perfectly healthy, healthier than their physicians, and do well. So what is it that drives this individual difference? I'm almost done. So what about the heterogeneity in these biomarkers of comorbidity risk? So this is very preliminary work from our cohort where we did a latent profile analysis of various biomarkers that we had measured. And this is not the whole cohort. And you see here that you can distinguish different profiles shown in the different colors for individuals in those cohort. The distribution, if you compare these profiles, how they are distributed between people with HIV and people without HIV, this slide, and again it's preliminary, you see that the distribution is different. The next step, and that's work in progress by one of my PhD students, is, of course, the question is belonging to a particular profile. Does that make you independently, does that independently affect your risk of certain comorbidity development? So that's work in progress, and as I said, an additional thing that needs to happen is can you relate these epigenetic aging biomarkers, for instance, to these profiles, and that's something that we are envisioning to try and do with our Swiss colleagues as part of that study that I showed you earlier. So I'd like to end, and also as an entry point to the next speakers, that this is all great science, and it will provide insight, and it may provide additional insight for general aging also outside of HIV. Clinically, of course, we're left at what do we do for our patients, and this is a consensus statement that also Rina, I think you were involved with, and on the importance of securing quality of life and going beyond viral suppression and making sure that our patients age and in a as healthy way as possible. And with that, I'd like to just acknowledge everybody involved in the cohort and our funders and turn the word back to the chair. Thank you.